today to our Yale Cancer Center Grand Rounds. Today is a special day. It's one of our endowed lectureships. This is the Blanche Tolman Lecture Series. It was established in 2012 by Marvin Sears, who many of you might remember was a longtime chairman of our Department of Ophthalmology and Visual Sciences. He established this series in honor of his mother, Blanche Tolman, who eventually succumbed to acute myelogenous leukemia. And this is the first endowed lecture series at Yale devoted to hematologic malignancies. It's intended to bring to Yale pioneers that have made major contributions to our understanding of the current trends in hematologic oncology. So we're very excited today to have Marcel van den Brink as our speaker. And to introduce Marcel today, I'm going to turn the podium over to Stephanie Helene, the director of our Division of Hematology. So Stephanie, the floor is yours.
Thank you, Dan. So it's my absolute pleasure to introduce Dr. Marcel van den Brink, who is the head of Division of Hematologic Malignancies at Memorial Sloan Kettering Cancer Center. So Dr. van den Brink is an expert in hematopoietic stem cell transplantation, and he obtained his MD and PhD from the University of Leiden, completed a postdoctoral fellowship at the Pittsburgh Cancer Institute, and his residency at Duke University Medical Center. He has been the head of the Division of Hematologic Malignancies since 2008, is also a professor of medicine and immunology at Weill Cornell Medical College. As a physician scientist, Dr. van den Brink studies allogeneic stem cell transplantation, both in the clinic and the laboratory. And his research is currently focused on two areas. One is to study the role that microorganisms living in the intestine play in patients undergoing stem cell transplantation and in those receiving cancer immunotherapy. He's developing strategies to help the body rebuild the immune system after bone marrow transplantation. His research in both of these areas is being translated into clinical trials that are currently ongoing at Sloan Kettering and beyond. In 2010, Dr. van den Brink started the Susan and Peter Solomon Divisional Genomics program, which focuses on targeted therapy approaches for patients with blood cancer, such as leukemia. And this program was actually instrumental in the development of the first genomic profiling test for hematological malignancies called Foundation One Heme, which we are happy to use. So I'm turning over the podium to Dr. van den Brink, who will tell us incredibly exciting stories on the intestinal microbiome and stem cell transplantation. So welcome, and we look forward to your presentation.
Thank you so much. Thank you so much for these kind words. Of course, this is again going to be a lecture by Zoom, and we were just saying how slowly but steadily we're getting a little bit tired of that and would like to have some real physical lectures again and see your audience and talk with your audience. But no matter what, it is a fantastic honor to be your guest and to speak for you. So the first thing that I have to tell you, honestly, is that I do have a conflict of interest because some of the data, some of the studies that I will be showing were actually sponsored by the company Cirrus. I'm not sure if I still need to show these kind of slides. I think that most of us will have a concept now that the microbiome that lives inside of us and on us is definitely relevant for a lot of the physiology. And one way of looking at that is what is summarized here, that one should start thinking about a human multi-species symbiotic supraorganism with a constant interaction between microbes and human cells. So we've been doing that basically since 2009, and our focus was very much when we started on allogeneic transplant patients, but since then we have broadened our whole scope. These are the current leaders of our group, and the original gangster, Eric Paymer, has since then left us and has bolted for the University of Chicago. So these are the folks within my lab that are working on this, and I will mention some of their names when we get to their studies. So an easy way to summarize about 10 years of work by our group and by others within the context of allogeneic bone marrow transplantation and trying to see if there's a clinical relevance to changes within the gut flora is to take as a starting point the causes of death within the first year after allogeneic transplant. And if you do that, then you can paint a cladogram and indicate with a blue color taxa that are linked to good outcomes and with a red color taxa that are linked with bad outcomes. And you can differentiate the various clinically relevant outcomes. First and most of all, of course, the overall survival rate, where you can see that certain taxa are linked in a positive or a negative way. Then, of course, the second one that we have focused on very much is if certain taxa are linked to a graft versus host. And I'm giving you one study here from 2015, where we demonstrated in patients that during the time period of neutrophil engraftment, which is about 14 days out from allogeneic transplant, that the abundance of a commensal anaerobe called Blaudia was clinically relevant, it seemed, because patients who at that point had low levels of that taxa had a greater incidence of lethal graft versus host, which is marked here with these red bars, and that led to overall worse outcomes. Many other clinically relevant outcomes can also be linked to changes within the flora, such as infections, organ toxicity, and even a relapse. So now you can start to paint a picture really of what the different taxa could be linked to. And in some cases, we have some mechanism also, and I will show you some of that. Now, many of these studies that we did and that others did were limited by small group sizes. For instance, here I am showing a study early on where we demonstrated that if you look again at the time of neutrophil engraftment after an allogeneic transplant, at the diversity within the gut flora, that patients who at that point had higher diversity had a better overall outcome, and that seemed to be linked to the incidence of lethal graft versus host. But again, this was a single center, small study, so we felt very fortunate when some of our dear friends and colleagues from all over the world were willing to work with us, so that now we could do a much larger study looking at 1,300 plus patients. These patients were getting allogeneic transplants for AML, MDS, NHL. And the first thing that really struck us that if we looked at the baseline samples, so the samples when patients come in for their allogeneic transplant, that the composition of the flora was not that different between those centers. And I'll give you some reasons for that later. And second, that in all four centers, what we notice is that the moment that they come in for allogeneic transplant, there is a dramatic drop within the diversity of the gut flora. And does that matter clinically? Yes, it does, as I'm showing here. Again, taking as a time point around neutrophil engraftment, which, as I said already, is about 14 days out from allogeneic transplant. Patients who at that point had higher diversity had better overall outcomes, and this was holding up for the New York patients, but also for the combined cohort of the other three centers. When we took a deeper dive, so what makes or what leads to that difference, then it seemed to be mostly linked to transplant-related mortality, not so much relapse. And within that category, it actually seems to be mostly a difference in lethal graft versus host again. You can go one step further and you can start to think about certain taxa that are linked to more favorable or worse outcomes, and that you can validate then again by taking all of the patients of the other three cohorts and indeed see that certain consortia would be linked to better or worse outcomes. As you can tell, we're not so focused on really zooming in too much on certain taxa except for one, and I'll get back to that one later. I told you already that these patients came in with fairly similar diversity and the composition of their flora. And when we analyzed that actually against normal healthy folks, what we saw is that at all of these centers, patients come into a transplant with lower diversity. And we speculate that that is because most of them will have gone through a year or so of chemotherapy, neutropenic fevers treated with all kinds of antibiotics, and so on. But what was interesting is that coming in with an even lower diversity, coming into a transplant, was again linked to worse outcomes. Similar findings we have now also for Ottawa Transplant, where we see a similar drop within the diversity, which starts at the moment that these patients come in for a transplant. And again, if we take as a mark the time point of neutrophil engraftment, which is about 9 days out from Ottawa Transplant, we see again that having at that time point higher diversity is linked to better overall outcomes. Now, meanwhile, a number of studies specifically within checkpoint or blockade have also demonstrated that diversity seems to matter for certain outcomes, in this case, responses to checkpoint blockade. And we have some early data also that this might matter for the efficacy of CAR cell therapy. Some studies that we're still finishing at the moment seem to indicate also that changes within the gut flora, specific taxa, could be linked with the pace of the CD4 regeneration after an allogeneic transplant. I don't want to make too much of a deal here of these various taxa because we still want to take that into a germ-free mouse models and study that further. But this gives us hints of which cell types might be, or which taxa might be relevant. Another critical feature is that with the loss of diversity, what happens also is that in some of these patients, specifically within the post-transplant period, there is a moment that their whole flora is being dominated by a single taxa. If you use as a definition that domination is when more than 1/3 of your flora is dominated by a certain taxa, then we actually notice that in all patients at all centers, they will have at a certain time point a dominance, or almost all. And what was very striking is that all four centers, that the most prominent bacteria that would do that is Enterococcus. And we knew already from studies at our center that having a state of dominance with Enterococcus within the post-transplant period was linked to a 9-fold risk for bacteremia with VRE, for instance. But what was very striking is that at all four centers, it was one specific species that would do that, that would lead to a state of dominance, and that was Enterococcus faecium, as I'm showing you here. And that seemed to matter clinically also, because what we noticed is having during the period or the post-transplant period at one point a state of dominance with Enterococcus faecium was linked to greater risk of a graft versus host, worse overall outcomes, and specifically an increased incidence of lethal graft versus host. That was true for all of the New York patients and also held up when we took the 3 cohorts from the other centers together. So we took that into mouse models, and what I'm showing you here is every box is one mouse where we did sequential sequencing. And in this case here, if we add some T cells to the allograft with which these mice were being transplanted, which will lead to a graft versus host, as you can see here, lethal graft versus host, then you must notice that there are these red diamonds here, which means that there's a blooming of Enterococcus happening during the development of a graft versus host in these mice. These mice are not getting any type of antibiotic or anything else. We thought first, well, maybe that is just for one strain or for one setting. So we did different strains and 3 different settings for monitoring a graft versus host, causing a graft versus host. In all of these cases, we kept on finding about 7 days out from allogeneic transplant, during the development of a graft versus host, there is a blooming of Enterococcus. Does that matter in these mouse models? Well, we tested that by taking a germ-free mice, giving them a minimal flora plus or minus Enterococcus. In these mouse models, by the way, we saw blooming with a different species, was not faecium, but Enterococcus faecalis. And if we did that, if these mice had Enterococcus in their flora, then indeed they had worse graft versus host, and again had a blooming of Enterococcus. So we took that further into these mouse models and analyzed mechanisms. And since this is published, I'm only going to summarize it here with some schematics. So what we think is happening and what kind of data we have so far is that the damage caused by chemo and by the conditioning regimens plus the alloactivated T cells, which specifically target the crypt stem cells and causing a graft versus host within the gut, that will lead to enterocyte damage. The enterocytes, therefore, start to make less of an antimicrobial protein called REG3, which is known, as we and others have actually demonstrated, to be an antimicrobial protein that can contain Enterococcus. Another thing that also happens is that the enterocytes, specifically within the ileum, are less capable of making a lactase. That will lead then to increased levels within the lumen of lactose. And that, plus the fact that there's less of REG3, will then lead to an Enterococcus bloom. The Enterococcus bloom pushes away some of the commensal flora. Well, one of the beneficial things that the commensal flora does, as we and others have demonstrated, is that it makes a butyrate. And butyrate is an intraluminal nutrient for these enterocytes. So if there's less butyrate, then that will lead to even more damage to the enterocytes. And now you're in a downward spiral and things get worse and worse. So we were trying to figure out, are there ways that we can maybe block that? And we thought initially about some bacteriophages and other things. But then the postdoc who was working on this, Christoph Stein-Thüringer, did a very simple thing. He analyzed simply what are the pathways, what are the nutrients, as I mentioned already, that Enterococcus favors. Well, as I said already, it likes lactose. So in his culture system for Enterococcus, he simply poured some lactate from the local pharmacy and demonstrated that with that, of course, he could block the growth of these bacteria. He then went back to these mouse models, and what he did there, he's bought chow without lactose, which is actually difficult because lactose is everywhere in many different nutrients. But he was able to get that made, and when he put these mice in 2 different models on a chow that was lactose-free, he could get somewhat less graft versus host. I mean, you're not curing a graft versus host with this. And he could block the blooming of Enterococcus. So then he took that finding back to humans. And we looked in our patient cohort, are there maybe patients who have lactose intolerance? When we looked at that, what we hoped, of course, is that that would be linked to increased levels of a graft versus host. We didn't really find that. There was a trend. But what we did notice is the moment that patients come off antibiotics, and that is the 0 here, then those patients who are lactose intolerant will have higher levels of Enterococcus. So as I've been trying to show you here in this part, so the Enterococcus can dominate in the post-transplant period. It is linked to a graft versus host, and lactose is one of the basic nutrients for Enterococcus, and using lactate or basically strategies like that could to potentially block the bloom of Enterococcus and in that way limit the graft versus host. This of course begs for a clinical study, which we haven't done yet, so I can't tell you anything about that. Meanwhile, other centers have also demonstrated that the levels of Enterococcus within the post-transplant period are linked to a graft versus host. I'm just showing you one out of several here. Another disease that we were interested in is the complication of chronic graft versus host after allogeneic transplant. So we tried to figure out if changes within the flora could be relevant for that also. Now, the onset of chronic graft versus host is, of course, much, much later. It's about 200 days out. And what we did in this case is we looked at samples about 100 days out and tried to see if there were certain taxa maybe that could be linked in a favorable or an unfavorable way with the onset of chronic graft versus host much, much later. And indeed, we have some hints now, such as Streptococcus and Akkermansia, that seems to favor the onset of chronic graft versus host. So of course, this needs much, much more work. Another feature in this article that I'm not summarizing is that there might be also a role for certain short-chain fatty acids that might limit the incidence of chronic graft versus host. Now, I mentioned already a few times that this drop in the diversity within the gut flora is pretty dramatic in all patients who have an allogeneic bone marrow transplantation. So we went back and we tried to analyze what are possible factors that might cause that dramatic loss. And the first one, of course, that we looked at was antibiotics. And indeed, if you look at the use of broad spectrum antibiotics, so those type of antibiotics that we typically give when a patient has fever and neutropenia, and specifically will do damage to the commensal anaerobe flora, then indeed the exposure to those types of antibiotics will lead to a greater drop in the diversity. We analyzed over a period of about 10 years the use of antibiotics in our allogeneic transplant patients and try to see if certain types of antibiotics were linked to greater incidence of lethal graft versus host. And we came up with two, piperacillin tazobactam and imipenem, also meropenem as we're using it now, but for this study we could only look at imipenem. And those are indeed two types of antibiotics that do more damage to the commensal anaerobe flora than many other types of broad spectrum antibiotics such as acefapine. As I'm showing you here, that was linked indeed to higher incidence of lethal graft versus host. We took that again into a mouse model, and we could see indeed that these two broad spectrum antibiotics that damage the anaerobe flora would lead to worse graft versus host. And to make a long story short, because this is all published, when we studied this further in this mouse model, we saw a few things. First of all, we saw a change within the gut flora, that there was a blooming of a bacteria, Akkermansia, and Akkermansia lives very close to the mucus layer and has mucolytic enzymes, and therefore will lead to a greater breakdown, we actually speculate, of the mucus layer. And we could demonstrate that the gut barrier function was more impaired in these mice treated with this broad spectrum antibiotic than not. And that again might set up a cascade of a number of things, more stimulation potentially of certain dendritic cells that I won't get into now. They will make higher levels of a cytokine that we know is linked to gut graft versus host, which is IL-23. That will lead to greater activation of allo-activated CD4 T cells in this model that we are using. Those are the driving donor T cells that will give you a graft versus host, leading to worse overall graft versus host, specifically within the colon. So a number of studies have looked now over the last decades or so if the use of broad spectrum antibiotics has any impact on outcomes after allogeneic transplant. And as we were chatting earlier, the first studies looking at the use of antibiotics, broad spectrum antibiotics in humans in the 1970s and 80s actually seemed to indicate that wiping out the whole flora would lead to better outcomes, specifically less graft versus host. And there are some pediatric studies that still seem to indicate that. But the bulk of the stories, the bulk of the studies over the last years do seem to indicate that the use of broad spectrum antibiotics is linked to worse outcomes, specifically increased levels of lethal graft versus host or graft versus host overall. So what can we do about that? Well, one of the things that we have been looking at is a beta-lactamase that you would give orally, so that within the lumen you can block any kind of effects of whatever type of antibiotic you are using. So these are some early studies with such a compound. So if we give that, then indeed we can block in a normal mouse the change within the diversity, the blooming of Enterococcus, when you treat a mouse with both an antibiotic and this beta-lactamase. And if you take it to a mouse model for a graft versus host, similarly, you can somewhat block the worsening of graft versus host that you would get with the antibiotics. So of course, we're looking forward to taking this into trials now. A second major factor we think that can impact on the dramatic loss of diversity are the different types of conditioning regimens. So we took a deep dive here at all of the different types of conditioning regimens that we have been using at our center. And as you can see, there are many. You can put them into three categories based upon their strength, going from myeloablative to a reduced intensity to non-myeloablative. And if you do that, as you would expect, the ones with lower strength indeed give you less of a drop in the diversity. And even if you control for the use of antibiotics, you still keep on finding that same thing. Another thing that was very interesting when we looked at it in some more detail is that certain regimens, and we don't know why, we need to study that further, such as this one with fludarabine, cyclophosphamide, and low-dose TBI, seem to be linked to the blooming of certain bacteria. And here I'm pointing out again the one that I mentioned earlier, Akkermansia. Another factor that hasn't been studied with that much detail yet, but we know is a major factor for changes within the flora, is diet. So to be able to get accurate diet data, we hired a nutritionist who very carefully, day by day, in almost 100 patients, monitored exactly what these patients ate. The first thing that he noticed is if he looked at the onset when patients come into transplant, that calculating the nutrition risk index, patients coming in with lower levels for that index have already a lower diversity within their flora. Another thing that he noticed is that the calorie intake, the moment that these patients come in, goes down dramatically, and it follows sort of the same pattern as that drop within diversity. And he first sort of analyzed the usual aspects that people look at when they're studying a diet, so calories, protein, fats, fiber, and so on. And he found indeed that calorie intake was positively correlated with diversity, fiber also, and also positively with Blaudia. And that is true for both calories and fiber, and negatively for Enterococcus. So that was interesting. But what I actually found even more interesting is a different way to look at the diet, and that is to look at it as a taxonomy. So now you look at all of the food products more than these categories like protein, fat, and so on. And when we analyzed our data like that, what we saw was that the food diversity immediately dropped when patients come into a hospital, and that diversity again drops more for those patients who get a stronger type of a conditioning regimen. You can then start to look at certain food groups and how they are linked to a diversity. And that was very interesting because then you find something that we didn't really thought of, and that is that the intake of fruits and sugars and sweets and beverages, that is actually linked negatively to the diversity. So we're still trying to figure out why that is. And one of the theories that we have is that these sugars, these very simple sugars, that they actually might feed some of the pathogens or the bacteria that are taking over in times of low diversity, and in that case might make matters worse, as has been shown, for instance, for an enteritis, for a colitis model. And again, you see there, if you feed these mice while they're getting DSS also simple sugars, then you see a blossoming of, again, a bug like Akkermansia. So now we can start to make these kind of tables where we can see what food groups might have impact on certain taxa. And of course, this can help us to start to compose maybe a diet that would be beneficial for specific patients in specific settings. And that is, of course, our ultimate goal with all of this. Another category is drugs. And patients who are getting an allogeneic bone marrow transplantation at any given moment are probably on 7 or 8 different drugs. And there was a very nice study a couple of years back where it was demonstrated that many drugs that weren't antibiotics, that they actually could also impact on many of the bacteria that are part of the commensal flora. And just to highlight some of these drugs, these are all drugs that we frequently give to our patients, including things like cyclosporin. So a very talented graduate student did the following thing. She took all of the data that we have from all of the samples on 1,100 patients. She put them in a UMAP and therefore could see all these clusters. Then she analyzed these clusters a little bit better. She came up with 10 different clusters and labeled them. And then analyzed, since we had kinetic data, if the starting or the stopping of a certain drug would have impact on the flora in these patients moving from one cluster to another cluster or staying put in that same cluster. And when she did that kind of an analysis, what was very striking is that, of course, the antibiotics will have impact if you transition to another cluster or if you stay where you are, as you can see here from this data. But all of these other drugs, and she looked at a grand total of 63 different drugs, can have impact also. So it's a little bit early to show you data yet, but we have some data now that seem to indicate that certain pain medicines might have impact on changes within the gut flora. So there's a lot of work still there that we can expand on. Now, of course, the ultimate goal for many people is to take this back into the clinic. And we've been thinking, of course, about that also. I'm still very cautious because I feel that we're in the early goings, that we still need to know much, much more. But if you categorize the different therapies in 4, then you can think about the use of antibiotics, and that is probably the lowest hanging fruit because those are drugs that we give and that we can easily monitor. A second category would be prebiotics. What we're thinking of there is to maybe give specific nutrients that would help, that would feed, that would favor taxa that we think could be of benefit. The one that most people are focused on is probiotics. So now we're talking about the fecal transplant, engineered microbes, and so on and so on. And there's certainly within allogeneic transplant, there's a lot of work going on within that field. And then a fourth category would be postbiotics. So those could be certain products made by bacteria. I mentioned already short-chain fatty acids, such as butyrate, and there are trials going on with that. What are we doing at the moment? Well, as I said already, for us, the lowest hanging fruit is antibiotic stewardship. Avoid the use as much as possible of these broad spectrum antibiotics that do damage to the commensal anaerobe flora. So we have a trial open at the moment where patients who get fever neutropenia will be randomized to either getting our standard of care, which is piperacillin and tazobactam, versus cefepime, and try to wean these patients as quickly as possible off antibiotics. A second study that we have finished already is an autofecal transplant. So the thinking there was when patients come off antibiotics, which is about 14 days out from their allogeneic transplant, why don't we give them back their original flora from pre-transplant? And since this was led by Eric Paymer and Ying Tower, our primary focus was the prevention of C. diff. So we looked at that mostly. And as these things go in this series, the incidence of a C. diff was actually relatively low. So we didn't see much there. But what we did notice is, first of all, that the concept worked. You could indeed, this is the pre-transplant diversity pattern of a patient who then was transplanted again with an autofecal transplant, and indeed would get pretty much their own flora back. So the concept seemed to be working. But in terms of clinically relevant outcomes, the only thing that we saw in this very small series was actually something that we weren't counting on, and that is that the activation of certain viruses, which commonly happens within the context of allogeneic transplant, such as CMV and EBV, was somewhat lower in those patients who had been treated with an autofecal transplant. Another thing that we noticed is that autofecal transplants seem to favor the engraftment, reconstitution of neutrophils, lymphocytes, and monocytes. A study that we're working on that is not open yet is to really rationally design a consortia of these bacteria, pretty much based upon that wheel that I started out with, that whole cladogram where I indicated how certain flora elements were linked to good or bad outcomes. And based upon that, we have created a consortium, and we want to give these bacteria back again at that time point of neutrophil engraftment, which is about 14 days out from allogeneic transplant, as I've said many times by now. So with that, I would like to stop. I would like to summarize basically that what I've been trying to show you is that changes within the gut flora are linked to overall survival, lethal graft versus host, bacteremia, sepsis, engraftment, and even relapse. I gave you a specific story about how the dominance with Enterococcus within the post-transplant period is linked both in mouse and man to lethal graft versus host. And I told you about the various factors that we think can have impact on the gut flora, such as the use of antibiotics, but also other types of drugs, diet, and conditioning regimens. So with that, I would like to, of course, thank all of my funding agencies and my fantastic lab and the many folks who we have worked with at other centers. So with that, I would like to stop, and I should probably stop sharing also, if I can do that. It seems to be.
Well, thank you, Marcel, for this really, really fascinating talk. I have to say, I was coming up with questions, and with every next step, you answered my first question. So maybe I can start with one. So right, so you are receiving these patients for transplant after they have gone through months and months of treatment. And have you looked at how, for example, whether patients receive, azacitidine or targeted therapy or chemotherapy before coming to transplant? Does that affect what you see then in terms of transplant outcomes?
Yes, so this is of course where we still don't have very good data. We do have some collection also of samples from patients before transplant, specifically with AML. We see a bit of the same patterns, but it hasn't been analyzed that well yet, that we see with allogeneic bone marrow transplantation, that an AML patient getting an induction regimen will have the same pattern of the loss of a diversity, dominance with certain taxa, specifically with again, Enterococcus, but we need much more work to analyze that. And as I hinted at, almost every drug that they might have seen in the year prior to a transplant potentially could have impacted on their flora. So it's very worthwhile to look at that.
Okay, awesome. So we have questions from the audience, from Lohis Gouda, who says, great talk. And his first question is, how well does this correlate with amino acid, magic biomarkers, RAG3, ST2, etc.?
So as you know, since you know about these markers, then you know, of course, those are the markers that have been developed by Jamie Ferrara. And he is doing these kind of studies with Ernst Haller at the moment within the context of the MAGIC consortium. And I haven't seen direct connections yet between, for instance, which would be really interested, RAG3 gamma and flora. So those are the studies that they are doing, but I haven't seen any data from them yet. We have only very limited data because we haven't used that panel that they are using so much.
Okay, awesome. And I'm going to read you the second question from Louis, who's one of our transplanters. Is the New York Poop Bank commercialized for other sites to study?
The New York Stool Bank?
Yes.
Well, we don't have a New York Stool Bank. I wish actually that we have one. And the one that most people have used is Open-Biome. And I was just reading that they might have some trouble and that they are closing. And that is a company, a not-for-profit company in Boston. So that's where a lot of people have been getting flora from. We, at the moment, are working with some companies also, and I didn't put that into my slide, to potentially do a fecal transplant for graft versus host. And you might have seen very small series from all over the world where people have tried that for steroid refractory graft versus host. They would do a fecal transplant, different concept, sometimes that you just do a normal donor, or even one company is sponsoring a trial where they take a whole bunch of healthy folks and literally mix all of the feces and give one giant transplant with that. And they seem to have some benefit. So there is a lot of focus at the moment on doing fecal transplant for steroid refractory graft versus host, and with small series showing benefit, but we need much more work. And I want to emphasize that there are also risks, because as we all realize, you're dealing with patients where the gut barrier is negatively impacted by the conditioning regimen. So any kind of bacteria that you give there have a higher likelihood to pass the gut barrier, and you might know of the negative outcomes that we're seeing with some of these fecal transplants where the product wasn't carefully screened enough for certain bacteria, which led to 2 patients getting seriously ill and one of them dying. So there are a lot of risks there still.
Mm-hmm. Dan, you have a question. Do you want to ask it directly?
Hi, fantastic talk. Thank you. Do you see similar effects of the microbiome in auto transplants?
Yeah, so I showed some of that data. So for auto transplant, we see the same drop in the diversity, again, starting immediately. And we see also links to outcomes. So for instance, for myeloma, we could very nicely see that patients with less of a loss in their diversity would have better PFS and OS. So that seemed to be of real benefit. All of this needs to be studied in much more detail, because now, of course, you're talking about auto transplant, so you're not talking about a graft versus host and things like that. But there are signals there that are absolutely worthwhile studying further. Yeah,
awesome. I think it's fascinating that in this population you're studying the immune system so intricately, and can some of this work be transplanted, translated to patients who are not in the transplant setting in terms of, immune interaction? I think you were mentioning the effects on immunotherapy.
So I think that is, of course, where a number of companies, a number of centers, a number of scientists are going with this. The general concept being that the gut flora can modulate immunity. Which it almost has to, right? Because you're in a constant interaction there with the gut flora. So it's very clear that T cell repertoire activation of innate cells is very much modulated by changes within the flora. That is obvious. So people have taken this, of course, within the field of a checkpoint blockade much, much further. You might know there was a back-to-back science articles demonstrating that certain compositions of the flora were linked to better outcomes with checkpoint blockade for melanoma and so on. And that has led to a series of trials that are going on at the moment. It has also, and I always tell that story because I want to warn people, it has led to negative outcomes. And what I mean by that is that because so many patients heard about these stories, oh, you can do something with microbiome and my checkpoint therapy is going to go better. They went to their own pharmacy. They started to buy local probiotic drugs, etc. And a scientist at Anderson had actually carefully analyzed that and found that those people who did do it yourself probiotics had worse outcomes from their checkpoint blockade than patients who didn't do that. So there are certain dangers, and I think we have to warn people also about this. This is not sort of a free-for-all, and we still need to understand much more. What are the dietary elements? What are the bacteria that really matter for certain outcomes? As I illustrated also, simply telling people to eat a lot of fruit, well, in certain contexts, it might be a bad thing, actually. Who would have thought that?
Mm-hmm. Mm-hmm. Yeah, I'm just taking the questions. And it's fascinating that across centers, you know, in the world, whereas diet is probably quite different, that you have such homogeneous or similar starting populations.
Yeah, that we found very fascinating, right? I mean, you're talking with patients from Bayern versus the north of Japan, and you would really think the diets are completely different, and they will go into these transplants with completely different flora. But as I mentioned during my talk also, we really think that that is because most of these people have injured microbiomes to start with. They come into transplant already, having seen for a year or so many drugs and antibiotics. That is probably why it's so similar.
Okay, fascinating. So do you have a suggestion of a simple measure? So should we ask our hospital to change the diet, to change what food is served in the cafeteria?
Well, I think first of all, when we started to look at the diet, I don't know how it is at your center, but on our transplant floor, it's almost like an ICU, right? We have such detailed data about everything, vitals every 8 hours and daily chemistries and blood counts and everything. But when it comes to what do patients actually eat, most of what we saw is ate half a sandwich or something like that. So we have no detail about what people are actually eating. So I think that is a moment where we need to upgrade. We need to take that a little bit more serious. Now that we know that it's a major factor that can have impact on microbiome, and microbiome, I hope that you got that out of this lecture, really seems to impact on clinically relevant outcomes. So that's one of the things that I'm trying to fight for within our hospital also, that we take that a little bit more serious. We really need to know what our patients eat, not just nurses scribbling down like, well, ate something. And then we can learn a lot from it. And then we need to understand in much more detail which dietary elements do what.
Okay, that's fascinating. So I'm not getting any more questions, so I get to have all the questions and the entire conversation here for everybody. But you know, that seems like a fantastic project where you could potentially engage the patient, right, in documenting, using Epic, using well, maybe we'll close on that. Yeah. And maybe a fantastic collaboration that we could then do with you.
We'd love to do that with you.
Take the Epic interface and put it to use for patient care. That'd be wonderful.
Thank you.
Awesome. So we're not getting more questions. You've answered everybody's questions. So thank you so much again for giving a fantastic talk. And you certainly have my mind spinning. And I don't know if I should drink all my ginger tea now, but you know, see how that goes.
Probably okay.
All right.
Okay.
Thank you so much.
Thank you very much.
Thank you very much. It was great.
Thank you. Bye-bye.